today is Professor Kwanghun Chung. He's a joint professor in the chemical engineering department and also in the Dickower Institute in Brain and Cognitive Sciences, and also the department of IMS. I don't know, it's a complicated arrangement. But you'll see the work he's going to show you today is interesting to all those communities, and I think will be interesting to you as well. Professor Chung did his undergraduate work at Seoul National University. Then he did his PhD at Georgia Tech in chemical engineering, and then went to Stanford for a postdoc, where he invented some of the techniques I think he might show you today.
Thank you. I'm delighted to have this opportunity to share our research with you guys. So my lab is focused on developing and applying new technologies for studying complex biological systems like the brain. So human brain is probably one of the most complex systems in the universe. It consists of 80 billions of neurons, each making thousands of unique connections. And at this very moment when we discuss this topic, a subset of neurons within your brain are firing on a millisecond time scale. And that constantly changes your brain connectivity throughout your lifetime. And it's not surprising that this complex system often fails. 1 in 4 adults in US suffer from diagnosable psychiatric disorders like autism, schizophrenia, or depression. You may think that I'm mentally stable, I'm lucky, I'm not one of 4, but 1 in 3 of us will eventually get so 33% of us unfortunately get Alzheimer's disease. And this is how much we pay just for Alzheimer's disease. But unfortunately, there has been no major drug developed since 1970. And that tells us how little we know about our brain. We don't even know how many different cell types we have and how they are connected to each other. So it's like we have this kind of really crude, raw resolution old map, and we are trying to explore this the most complex universe and system with this old map. It would be ideal if we could have like a Google map that has all the information, that has all the connectivity, like the identity of buildings and road connections and etc. But unfortunately, we don't have satellites, we don't have drones, we don't have advanced transport system for developing human brain map. And we don't have those like enabling technologies when we study our brain. So I've been developing new technologies to address this issue. The first technology that I developed before I moved to MIT is called Clarity. And Clarity basically makes tissue which is not transparent. This is a mouse brain sitting on Lamoni card's code. And the Clarity technique turns this tissue into optically transparent and macromolecular permeable hydrogel while preserving its structure and molecular information. So it contains, even though it is transparent, it contains all the information. And because the brain is transparent, light can pass through the tissue without being scattered. And you can image the intact brain that contains all the connectivity without mechanical section, which has been the bottleneck in mapping the brain. So here is just one example. This is an image of a part of the mouse brain that I showed in the previous slide. This brain expresses fluorescent proteins in subset of neurons, about 5%, and this particular volume shows subcortical regions called thalamus, and this is brain region called hippocampus, where short-term memory is stored, and this is brain region called cortex, where long-term memory is stored. And because the continuity of these neural projections are preserved you can see like how these neurons are distributed in this volume and how they are kind of connected. A conventional way was, because tissue is not transparent, you have to make it, first slice it into thin sections, like 50 micron, and then image, and then do manual alignment, which is really laborious and time consuming, and you, many cases, use magnetic information. But with this technique, you can bypass sectioning and reconstruction and just see the entire. And here's another example. This is human brain tissue, prefrontal cortex. We got this tissue from an autistic patient, and we cleared it and labeled a subset of neurons called part1 positive neurons. And here you see these neural cell bodies and their neural branches, projections. And because the resolution is high enough, and because we can label these molecules and visualize the entire fibers, we can do reconstruction of individual neurons. And we found this interesting ladder-shaped interconnection between isoneural dendrites. So neuron has a lot of fibers. It has like big tree-like dendrite arbors and axons. And these dendritic arbors, they don't cross each other. They shouldn't make connections with each other. But here we found that some of these parallel positive neurons have these massive ladder-shaped interconnections, which clearly looks abnormal. So this technique is promising. You can get a lot of structural and molecular information. The problem is it's not scalable. The tissue that I processed here is only 500 micron thick. And we couldn't process tissues thicker than this because when you process large tissue, you have to control the chemical transport and kinetics. But the conventional way of processing tissue, you don't have that control. So what happened is, before chemicals diffuse into tissue, these chemicals react with molecules near the surface first. And the molecule, the tissue in the middle, basically don't have enough time or enough molecules to react. So the tissue processing is really highly heterogeneous. It's not uniform. So to address this issue, we developed at MIT, we developed a technique called switch. It's basically a way of controlling chemical interaction time and kinetics system-wide by using a set of buffers. So we have here two buffers. One is switch off buffer, shown here. That basically inhibits chemical reactions between endogenous biomolecules like proteins, DNA, RNAs, and exogenous molecules. Could be fixative, could be molecular probes, or any molecules. And we use this switch off buffer to first disperse an exogenous molecule throughout the tissue without any reaction. There's no reaction during this step. And once the chemicals are uniformly dispersed throughout the tissue, we move the sample to switch on buffer that basically accelerate the reaction. So that way we can synchronize chemical reactions throughout the tissue. So all the endogenous biomolecules within the tissue now can experience the same reaction condition, the same time and chemical concentration. That's really simple concept. You only need to use a set of buffers. And using this technique, we can first make tissue heat and chemical resistant. So you can do many rounds of staining, destaining, and imaging. And second, you can achieve more uniform staining. So I will show you some what we can do with this technique. So this is a human visual cortex, a small region of human visual cortex, layer 4-3. And here you see some of the neurons. labeled using SMI32 antibody and their projections. And there are a dark area, right? And you may think it's a like void volume, which is not. It's actually packed with many other cells and other tissue structures. And to study this human brain, right, or any complex biological systems, it is really important to understand the complex interactions among these many biological components, not just one cell type, but many, many types, and not just one molecule, but many, many molecules that each cell type uses. And using this technique, we can basically map out as many cell types or molecules as you want. So here we visualize carbamide positive cells, different neurotype. Carbamide positive cells, blue, carotene, yellow, neuropeptide Y, positive cell, the couple of them in this volume. And lectin visualizes all the blood vessels. So this is blood vessels. And GFAD visualize GFAD positive astrocyte, different cell type in the human brain. These are all the neurons, basically. And these are all the cell nuclei. So using this technique, we can basically get all this information from the same tissue in a fully integrated manner. And to do this, the first step is to make tissue hidden chemical resistant, because to label tissue multiple times, so what we did in the previous slide is we stain tissue with one set of molecular probes, image it, and then remove all the imaged molecular probes, and then do another round. And we did that 22 rounds, and then do correlation. And to do that, you have to destain, but you have to remove the molecular probes, and that requires harsh condition. You have to incubate tissue in a detergent solution at high temperature, like 70 Celsius. And normal tissues, they just get disintegrated immediately. So we had to first develop a way to make tissue hidden chemical resistant. And we thought that maybe we can use these multifunctional fixatives and increase intermolecular cross-linking degree to make tissue more rigid and more resistant against denaturation. So we tested these chemicals, and we found that, these chemicals, if you mix it with bovine serum albumin, which is protein, they make nice gel. And if you incubate these gels in the harsh destaining condition, the gel still maintain its structure, so it's stable. And next step is, okay, so we found these chemicals, right, that can make the biomolecule, they can form like stable gel with biomolecules, right? The next step is to deliver these chemicals uniformly throughout the tissue. So here we try the smallest chemical, which is GA, shown here, right? And what we did is we put a rat brain, which is pretty large, in intact rat brain in this GA fixative solution, incubate for 2 days, and then take out the brain, cut corner slices, and look at the structure. And as you can see, the outer layer is really dark, indicating that outer layer is very fixed and gel is very light. But the core part is white, meaning that there is not much fixation. There is no GA actually penetrate into this core region. And if you put this tissue, which is not uniformly fixed, in harsh destaining solution, the core just melts away, right? So we have to address this issue. And we thought, okay, maybe we can control the reaction kinetics between the fixative and endogenous biomolecules. And it's well known at low pH, endogenous biomolecules, the chemically reactive group become protonated, and they cannot really react with this fixative. So what we did is we incubate this tissue in fixative solution, but now at pH 3, which is switched on, right? And in this condition, fixative molecules can diffuse into the tissue uniformly without reacting with endogenous binding molecules. And once they are uniformly dispersed, we simply move the tissue to pitch 7, which is switch on button. And we did that, and we could achieve uniform fixation gel formation, and we put it in the same harsh descending buffer, but now everything is in there. Okay, so now we can uniformly preserve large-scale tissue. And we check whether protein is well preserved, and it turns out protein loss is around 3 to 4%, which is a lot better than clarity. We also check if the epitopes, so if this switch fixed tissue can be labeled with conventional antibodies. And we check 90 antibodies, and it turns out 86 out of 90 are compatible with switch fixed tissue. We also check whether structure is well preserved. So this is clarity technique after harsh destaining, and you can see that this 1 millimeter thick tissue, by the way, this is the cross section, and it was 1 millimeter thick uniform, but after the harsh destaining step, it kind of collapsed. The tissue kind of collapsed, and the thickness is not uniform, right? Indicating that we are losing macroscopic structure information. Whereas switch, after the same treatment, the overall shape is well preserved. And then we check microscopic structures. So this is 2 neurons expressing GFP before any treatment. And this is after switch-mediated gel formation, clearing, and staining. And as you can see, the microscopic structure of the neurons are very preserved. So using this technique, we were able to label this human visual cortex tissue over 22 times without losing the tissue information. And we collaborate with Sebastian Sung at Princeton and Medforce at MGH to develop co-registration algorithm to co-register all these data sets at single cell resolution. And we used blood vessels as a landmark. And here to prove that we don't lose structural and molecular information, we did high-volume staining 2 times. First staining for round 7, and this is round 19. So there were total 22 rounds of staining, imaging, destaining between these 2 rounds. But even after that, as you can see, we could achieve 99% co-registration accuracy, indicating that we are not losing structural and molecular. So now, after like 22 rounds of labeling, imaging, co-registration, now you have this rich data set that contains a lot of information, fully integrated. And with this data set, you can basically do high-dimensional quantitative phenotyping of the human clinical sample. So these are the analysis that we did. I will highlight just a couple of them. So it's hard to see, but here we visualize many different cell types. You can see the pattern and etc. And you can also extract morphological information of these cells. So this visualize cell body size of new and positive cells. And you can do unbiased combinatorial protein expression analysis. So basically here in this analysis, we used 6 proteins, 6 protein markers, right? And with 6 proteins, you have total 63 positive combinations. Right? And you can just look at all of these possible combinations and get quantitative information in an unbiased way. And we observed some interesting neurons. So it turns out there are many neurons expressing this By the way, these are calcium binding proteins expressed in a subset of those, carbine, carbapenem, carbapenem. And there are many cells expressing these two specific calcium binding proteins, carbine, carbapenem, or carbine, carbapenem. But there is no cell expressing carbapenem, carbapenem for some unknown reason. We don't know why. And also, we found that many of these calcium binding expressing proteins do not express UN, which is where a widely used pan-neuronal marker. So people use UN to visualize all the neurons, and they think, okay, UN is expressed in all cells. But it turns out, like 30% of these carbine expressing neurons, for some reason, do not express UN. Also, we could characterize the distance between cells and blood vessels. It turns out pretty much all the cells are within 20 micron range from the nearest blood vessel, which is quite amazing. It's just it's one cell body distance. And we could also characterize their the distance patterns. So basically this shows that we can do a lot of quantitative analysis using this technique. And it's scalable to large tissues because the tissue is heat and chemical resistant. You can clear them, make them transparent passively at high temperature. And it doesn't take long. Like in case of mouse brain, it takes about a week. And human brain or even marmoset brain, like small monkey brain, you can clear within a week. Another advantage is this technique preserves a subset of lipids. Lipids like fat-like molecule that basically makes lipid bilayers, like the cellular membranes. And we can use lipophilic dyes to visualize myelinated fibers, that has like high concentration of these lipid molecules. And when we try to label sick tissue using this method, right? When we try to stain all the myelinated fibers in this sick tissue using this lipophilic dye, we couldn't achieve uniform staining, right? What happened is, so this is a tissue, 1 millimeter thick. And what happened is the dyes go in, dye molecules go in, and they get consumed by all the target molecules, highly abundant target molecules along the outer surface. That's why you don't have any staining. So we decided, okay, maybe we can try switch concept. So we first screened buffers that inhibit binding. And we found that okay, if you have 10 millimole SDS, you can effectively inhibit this dye and myelinated fiber binding. So we use this PBS buffer that contains 10 millimole SDS as off buffer. And then we use PBS without SDS as an on buffer. So what we did, we first incubate tissue in off buffer containing dye. The dye diffused into the tissue uniformly. And then we simply move this tissue to on buffer. And using this simple switch approach, we were able to achieve this uniform staining. So this movie shows the labeled fibers. So here you see the fibers running. This is bundle of fibers. This is brain region called striatum, brain region called cortex. And it's extremely difficult to visualize all these myelinated fibers because they are everywhere. They are so many. If you use antibody that labels these fibers, you have to pour a ton of antibody. So it's not really practical. But using this switch concept and using this cheap lipophilic dye, we were able to uniformly label this sick tissue. And with this data set, so this is the 3D rendering of the data set. And with this data set, we try to see if there's any odors in this fiber geometry. And there is a paper published in Science in 2013 claiming that all the fibers have like grid structure. They are not totally random. They have an X they follow X, Y, and Z coordinate. So the paper claims that there is like a highway system in our brain. And we try to check that hypothesis using this data set. And it turns out, really, you can really see fibers running like this way and this way in this plane. It's hard to see because of the projector, but there are two fibers running this way. And if you look at this orthogonal plane, there is another set of fibers running like this. Can you guys see? So indeed, there are fibers running like X, Y, and Z. And we thought, okay, maybe we are trying to see what we want to see. Right? There might be some other fibers running randomly. So we did unbiased autocorrelation analysis to really capture this fiber orientation, and we found that indeed these fibers make 90-degree cross sections. And this switch concept is also scalable to larger tissue. We labeled whole hemisphere, which is much larger than the 1 millimeter thick tissue that I showed. During the switch concept, here basically we just increase the incubation time. So we incubated this tissue in switch off buffer for 4 days, and then move it to switch on buffer for 2 days. And we were able to label all the fibers within this large hemisphere. And this tissue was imaged using spin, which Hitchin developed, and we basically did it. So basically, by controlling the chemical reaction kinetics using a set of buffer, we can make this tissue processing scalable, tissue preservation scalable, and tissue labeling scalable. And here's another example. This is again mouse brain. It's Alzheimer's disease model, and we visualized A beta plaques that are known to cause neurodegeneration in Alzheimer's disease. So switch makes tissue processing scalable, but still it's really slow. We still rely on passive diffusion. In the case of human visual cortex tissue, it's only 100 micron, so you can do staining every 2 days. Just 1 day incubation is enough for antibodies to diffusing. But in this case, like one hemisphere, the mouse hemisphere, which is 6 millimeter thick, it takes 2 weeks. So let's say you want to do 10 rounds of labeling, then it will take minimum 5 months. Right? So to address this issue, to make this approach really high throughput, we developed another technique that we term stochastic electrotransport. Stochastic electrotransport is basically a way to speed up the migration of charged molecules. So you guys all know Brownian diffusion, Brownian random walk, right? Particles, they have some energy, they just move around randomly. And here we developed computational model to visualize the movement of Brownian random walk of molecular probes within nanoporous tissue. So this is just nanoporous tissue you can imagine. And these are pores, and black dots are basically wars. And we are seeing, you will see red dots showing up in the middle of the screen. And those are molecular probes. So here you see small dots moving around. And they don't move much, right? They just jiggle around, but they don't move much. Because the Brownian random walk in this dense nanoporous mesh is really slow. That's why passive diffusion of these antibodies, molecular probes are really slow. One way to speed up the transport of this molecular probe is applying electric field. So here we apply electric field across this screen. So you can imagine that there are two electrodes here generating electric field. And pretty much all the molecular probes are charged. So you can basically speed up their transport.